Hello everyone. I'm Ercüment Çiçek. I'm a faculty member at Bilkent University, currently visiting EPFL on my sabbatical. So this is a joint work with İlayda Beyreli and Oğuzhan Karakahya, who were graduate students in my group and graduated. So they moved on, and then I will be presenting today. They couldn't attend the conference. So the talk is about a gene risk prediction algorithm for comorbid neurodevelopmental disorders. So comorbidity means two conditions that frequently exist together. And today I'll focus on autism spectrum disorder and intellectual disability. So autism is a neurodevelopmental disorder characterized by repetitive behavior and impaired social and communication skills. Intellectual disability is characterized by below average intellectual function, like IQ less than 70. And they are pretty common. So autism occurs in 1 in 36 children in the States. So this is a very recent report. This was 1 in 54 or 56 a couple of years before. And similarly, intellectual disability affects like 1 to 3% of the population. So they are quite comorbid. 70% of the children with ASD also has ID. They share risk genes and copy number variation regions. I will show you a figure. And both are quite heterogeneous and oligo or polygenic disorders. So this figure from the Santosh Garajan group shows that these two disorders share a quite large number of copy number variation regions and quite large number of risk genes. So there has been a large-scale effort on discovering risk genes for both disorders. I'm showing you two papers that basically analyzed trios of thousands of patients to find exome-wide or genome-wide risk genes. And then so far we have been able to, for autism, discover around 100 risk genes. So the gene discovery is mainly centered around finding de novo loss of function mutations. So you have a healthy mom and a healthy dad, and you have a child with an uninherited mutation. So these parents, pass the mutational burden, which are below a certain threshold, but this child has a deleterious mutation which kind of, takes the phenotype above a certain threshold. For autism, currently it's a large puzzle for which we know roughly 100 risk genes. And then we expect or estimate to find another 900. But our current statistical power, despite this large cohorts, only makes us sure that this is like we were able to find 100 risk genes. So we want to know the remaining 900, and then we have roughly 20,000 pieces to choose from. So the research question we are trying to address is, can we use the guilt by association principle to pinpoint the rest of the risk genes using biological networks? So we formulate the problem as a node classification problem where the nodes are genes. And then this is basically a semi-supervised learning problem. So we have a graph of nodes where the nodes are genes. And then we know the label for a certain fraction, whereas the gray ones are unknown. So we want to train a model which is able to predict the labels for the rest of the genes. So there have been some work on this field starting from 2014, which all of these methods use a type of a biological network to solve this problem. And I will just briefly mention some of them. So the first one is Krishnan et al., a Nature Neuroscience paper from 2016. What they do is they train an evidence-based support vector machine. First, they create a list of ground truth genes with positive labels with varying levels of evidence of being a risk gene, and a non-mental health related gene list. And then they use a brain-specific function network to generate features for each gene. And then they train an SVM model to predict a genome-wide ranking for all genes to be a risk gene for autism. So this is a work from our group. In this work, we use the Steiner tree problem to find connected set of nodes. And for the first time, we were doing this in a spatiotemporal way. So we were using multiple gene interaction networks this time, and then try to inform the model with respect to varying edge connections through time and space. And the final one is for ASD that I want to mention. So this one basically collects all information from the literature about these risk genes and then gets the same or similar evidence levels for the ground truth genes and basically trains a random forest model. One problem with this approach is that once you use these methods, which also rely on the labels that you use to train your model, there's a possibility of leaking some information, so we want to avoid that. So the shortcomings of the literature is that they are by design limited to work with a single disorder. So they all analyze autism or intellectual disability. So if you want to do this together, you either bag all the mutational burden for each gene and treat each disorder as the two disorders as the same, in which case you lose the disorder-specific information, or you perform an independent analysis per disorder. But then you have less mutation burden signal, and then your analysis basically lacks the statistical power. One other problem is that they usually use one or a few gene interaction networks. You either just pick one or you integrate them into a single one. In this case, you lose the information that's coming from each network. Maybe some of the networks are more informative, and then we had seen that multiple networks help to solve this problem. So this is one other shortcoming. So usually, finding out which networks are important is a downstream task. So you find your genes that are interesting, and then you map them back to the network to try to see which ones are connected. So this is what's done in the Cell paper. They have an algorithm of kind to find the risk genes, and then they map them back to the networks and find which ones are having the most signal. But then basically you have an algorithm which is using this information. What if we could learn during the training period which networks are giving you more information? For this, we propose DeepND, which is the name of the algorithm. So we use graph convolutional neural networks to analyze not one but multiple gene interaction networks, as you see over here. And then we employ a mixture of experts model, which basically learns which one of these networks are giving the most signal to be able to predict the risk genes correctly. Finally, we employ a multitask learning approach. Basically, we are solving the problem for both ASD and ID in this case. And we employ some fully connected layers before so that the loss is affected by the loss of both disorders affect these layers. So we learn the shared component, but at the same time, the network-based analyses are independent from each other so that we also learn disorder-specific features. So the graph convolutional neural networks are kind of an extension of convolution so that they can work on arbitrarily structured data, unlike images. So we are using the Kipf-Welling approach, which uses an efficient propagation rule so that you can solve the problem fast. I'm not going to go into the details, but basically, so the embedding that you learn from the K-1 layer is multiplied by the transformation of the adjacency matrix and a matrix that you learn for this layer, for the connections. And then you add some nonlinearity like ReLU, and then that is going to be the embedding for the next layer. So that we utilize not one but multiple of them, each get the features for the genes, and then learns basically a risk for each gene. That's going to be input to the gating network, which is the mixture of experts-based approach. So multitask learning is a learning paradigm in which you have multiple learning tasks, and they by assumption, they have some shared components. So in this case, we know ASD and ID are related, so we expect to learn some shared information between them. So instead of learning two independent models, in which case you have less data and then more parameters to learn, have a model which shares some of the parameters, so learns the shared structure, but also some layers, some parameters which are independent per disorder. So this would be, so you have input 1 to output 1, you learn a single model for both tasks. Instead, the earlier layers share some information, and so the loss of the first task also affects these layers, as well as the loss of the second task affects these layers, whereas these layers upstream are independent from each other, so only the loss of one, for instance, affects these layers. So we learn some shared text. So in our case, so this graph convolution plus mixture of experts model are task-specific, whereas we have some fully connected layers which input gene-specific features, and these are affected by both the loss of both disorders. So the mixture of experts model is basically supposed to learn which interaction networks are more informative. So they also get the gene-specific features as input and assigns a weight to each output of each network, each convolutional neural network. And this is kind of a self-attention idea to learn the weights of each network. So the gene co-expression networks that we generate based on BrainSpan data set of Allen Brain Atlas. So they have a bulk RNA-seq data from 57 postmortem brains. So we construct 52 spatiotemporal gene interaction networks from here. They are co-expression networks throughout neurodevelopment, actually up to late adulthood. So we take windows from the neurodevelopment, generate the co-expression network. We have 13 time windows, and then 4 brain regions that are clustered together. So we have 52 gene interaction networks. So these were, so we are basically following the directions of the Cell paper to also have some type of benchmark against. So the features we use, we only use the PLI of the gene, so this model is not disease specific. So we use Simon Foundation's categorization of the risk genes on the high level risk genes, and then Krishnan et al.'s non-mental health genes. Similarly for ID, we dig through some review papers to find risk genes on ourselves and then have the same negative set. So going to the results, so we compare here with Krishnan et al. In terms of ROC and then AUPR. So gray is Krishnan et al. Purple is the single task version of our model, and then the pink is the multitask version. So this is the result for ID, this is the result for ASD. As you see, the pink one has also lower variance and also has a much higher median performance for both metrics, so showing the benefit of multitask learning approach. Here we compare so not all of the algorithms are comparable in terms of this cross-validation scheme. So here we get the final list of each algorithm and find the correlation of its final ranking with the ground truth genes. So we use a rank percentage threshold. So 20 means 20% of the top genes are considered positive and rest are negative. So how correlated this 20% is with the ground truth? And then we just vary that threshold. So it is better to be on top. Here you see with respect to the really high confidence ASD genes, our performance just dominates the others. And this is a relaxed set of ground truth genes. Same for ID. And this is the same analysis with respect to precision and recall curves. We also analyze which networks are more informative here. And we basically regenerate the same platform that Cell paper. So we confirm some of the findings of those and now find even the later periods could also be very informative. So we basically kind of unite this network analysis into our approach. So we actually we kind of confirmed this. We checked how connected the ground truth genes in these networks, and we find that one of our top predictions has like 75x more connections compared to a network which is not that informative. Here we pinpoint a specific gene which has a very low prior signal, but we rank it really highly for autism in a protein interaction network which we don't use. It turns out to be HAP, so kind of verifying our results. So we had some unanticipated novel predictions. So this gene, for instance, a transcription repressor, it has a higher ranking for in Krishnan et al. It is quite low ranking for ASD, but we have a higher chance for ID. So and then the literature kind of supports this because this is associated with a syndrome that's for which ID is a comorbid disease. I can skip this. Here are my conclusions. So DeepND is the first multitask gene risk discovery algorithm which can work on comorbid disorders, can utilize multiple networks and learn which networks are quite informative. And so this is not specific to neurodevelopmental disorders. Actually, I didn't show you the results, but we ran some analysis on for ovarian and breast cancer, which are known to be comorbid, and then still this multitask approach is quite helpful. We also had some results on intellectual disability and ADHD. Again, multitask learning has improved the results. So we predicted also several novel genes for ASD and ID, and we hopefully have to dissect out the differences between these two disorders. So we published this paper in Patterns recently. You can reach the paper using this QR code as well as the code, which is available on GitHub. So I thank my graduate students and Simons Foundation for supporting this with an overseas grant. I thank all of our supporters, the funding agencies, and thank you for listening. I'm happy to take questions.